Start by thanking the conference organizers so much for having me here today. It has been such an amazing experience for me to interact with so many members of this variant effect mapping community via the Variant Effect Alliance, the Atlas of Variant Effects Alliance, and then also our additional collaborations that I'll talk about today. So I am a cardiologist at Stanford University, where I see mainly patients, adult patients, who have inherited genetic cardiomyopathies and arrhythmias. And so that's really what brought me to the space of trying to understand how variants can affect function and ultimately cause significant disease risk. So when we see a patient in cardiology clinic, it's similar, I think, with respect to how we think about using genetics in cancer, except that what we're talking about is a different disease endpoint. So for our cardiomyopathy patients, the idea is that their natural history really starts with them inheriting a causative variant or a variant that confers significant risk of developing cardiomyopathy, that they then can have genetic diagnosis early on in their course, which may enable us to prevent their disease, as it can with familial hypercholesterolemia from leading to bad outcomes. Then mostly for patients with cardiomyopathy, they have to progress to having symptoms or other incidental findings that lead to their diagnosis, at which point we hope that we can employ a disease-modifying therapy. And in cardiomyopathy, we are getting closer and closer to being able to do that on a precision basis based on genetic diagnosis. And what we're trying to prevent then is these outcomes of sudden death or the need for an LVAD or heart transplant, which of course are really life-altering and population-level problems in our society. So as we have covered throughout yesterday's format as well as today, VUS really are a major problem for us. And so what we're going to talk about today is how we're trying to address VUS with variant effect mapping in cardiomyopathy. This is some cardiovascular medicine specific data simply showing us that as we increase the number of genes that we test on our clinical panel for cardiovascular disease, we increase, as you can see, the preponderance of uncertain significance tests that come back here in black. And we decrease benignity, but we really only minorly increase our diagnostic tests. So before we launch into how we are trying to model cardiomyopathy for variant effect mapping, I think it's important to take a step back and think a little bit about the cardiomyocyte and the mechanisms that cause inherited cardiomyopathies and arrhythmias. This is a schematic that shows you the functions of many of the genes that are involved in these diseases. Some of them, as we've talked about, overlap with other disease spectra. For example, lamin AC, which is of course a ubiquitous nuclear envelope protein. But many of them are very specific to the cardiomyocyte and to its function. Those in particular are centered here at the sarcomere, which is the molecular motor of the cardiomyocyte, and can lead to diseases like hypertrophic cardiomyopathy as well as dilated cardiomyopathy. And then also here in the desmosome, which of course exists in other epithelial cells, but have different functions, I think, as far as conferring electrical signals between cardiomyocytes. So our major goals when we started out on this journey of variant effect mapping for inherited cardiomyopathy were, one, to see if we could identify high-level single-cell phenotypes that could be deployed across genetic causes of cardiomyopathy in order to really create high-level variant effect maps across diseases in our specialty, and then also to use those variant effect maps to illuminate variant-specific disease mechanisms. As many have pointed out today, there are genes that have pleiotropic consequences, but there are also variants within specific genes that have different consequences as well. So what we're going to start by talking about is sort of what we know about how to use variant effect mapping in cardiovascular disease. And I think it's important to note that, of course, our plenary speaker today, Fritz Roth, and his team, Daniel and Atina, who are both here as well, have really led the charge in bringing variant effect mapping to the cardiovascular space. This is an example of a collaboration that we all have together through the CardioVer Consortium, which includes as well Andrew Glazer, Dan Roden, Callum McRae, and Ewan Ashley. And what we did is we took this really great map of calmodulin that Fritz and Jochen had made, and we applied it clinically to several cases of pediatric sudden cardiac arrest. And we're actually able to verify the pathogenicity of 2 variants, as well as to show that a variant that had been de novo actually might not be enough in that family to exclude diagnosis based on its functionality. So the process and the outline for the presentation that I'm going to talk about today for modeling cardiomyopathies with variant effect mapping is summarized here. And I know everyone is very familiar with this pathway here, but I wanted to point out that the terminology that I'm going to use today was put together in really a massive effort by the ETS workstream of the Atlas of Variant Effects Consortium, really led by Melina Klauschnitzer, my co-chair at the time, Laura Muffley, who's been instrumental for everything we do in the Alliance, and then Benny and Alan, who are now co-chairing the ETS workstream, and then a great and huge thanks to Alan Alan Rubin, who leads the DCD workstream, and putting together a controlled vocabulary that we can use to describe the science that we do. So I'm going to start here by talking about how we are generating our variant libraries and in what cellular context we are doing so. I think it's important to start with the cellular context because, as I mentioned to you, many of the genes that we're interested in modeling do, of course, have specific functions in cardiomyocytes. And so therefore, we wanted to use induced pluripotent stem cells, which, as many of you know, can be reprogrammed into cardiomyocyte and phenotype here. These take about 3 to 4 months to generate from a patient, but once you have a line in a patient or a human that you believe not to carry significant risk of disease, they take about 30 days to differentiate into mature iPSC-derived cardiomyocytes. So why are we using this system, which is something that many of you who use these cells might ask yourselves on a daily basis, given how time-intensive they are? Well, first, as I mentioned, we are trying to model cardiomyocyte-specific genes, that those cardiomyocyte-specific genes have specific mechanisms and functional consequence in cardiomyocytes specifically, and that it's hard to find another cell type that really gets cardiomyopathy in that way. So in order to deliver a library into the cellular context, we had sort of 2 options. We thought the first was to integrate an in vitro generated construct, which could carry 1 variant per construct, or to potentially perform direct base editing at the native locus. Much of the work that I'm going to tell you about right now was done by Hannah de Jong. Her picture is going to be on the next slide, but she's here as well from Maze. We thought that the advantages of integrating the in vivo construct would be that we could control the number of variants that were delivered per cell, that we could actually saturate the region of interest with high fidelity, and that the pitfalls were that for many of these genes, the promoter really hasn't been worked out, and that allelic stoichiometry is actually quite critical in the cardiomyocyte to preserving a functional readout, and that it's very hard to deliver and ensure stable expression of large constructs in iPSCs. The advantages, of course, of direct editing at the native locus for us were that we didn't need to establish allelic stoichiometry for the reasons that were just mentioned, and that we could get stable expression where we wouldn't get the type of silencing because we were that we see with exogenous delivery in iPSCs. And then the pitfalls, of course, with direct editing are that saturation is going to be highly variable because it's going to be based on where we can place base editors using guide RNAs, and that in diploid cells, it's difficult to control the number of alleles that are affected per cardiomyocyte. So we initially, and by we I really mean Hannah, who led this work in collaboration with Fritz Roth's lab, decided that we wanted to generate those in vitro constructs with single variants. As you can see, Hannah was able to, using popcode from Jochen and Fritz's work, generate a really great and high fidelity library here. This is in myosin heavy chain 7. But then when we tried to deliver that library into cells using a dual integrated landing pad, we were really unsuccessful in getting the variant carrying constructs into these iPSCs using this method called DICE in any sort of high throughput manner. At the same time, Hannah and others in Ewan's lab were working together to try to get base editing up and running. We were using two different constructs. The first is CRISPR-X, which was developed in the Basic lab at Stanford and as a cytidine base editor. And then the second that we've recently employed is actually ABE8E from David Louie's lab. And we were actually able, with significant difficulty, to get these into iPSCs, which don't like to be infected by lentivirus, but it turns out will take electroporation of large plasmids along with many other small plasmids. And we were able actually to achieve pretty good mutagenesis here. So what I'm showing here is that this is 3 replicates of part of the RBM20 locus, is that we were able to get good adenine, cytidine, and G's and T's edited. We had, of course, a preference for the particular change that is usually associated with each one of the base editors we had. But using this, we were able to get high frequency of these replicates in high frequency of these variants in 3 replicates at reasonable frequencies. So having generated this library in the context of an iPSC, the next question that we wanted to tackle was how we could develop phenotypic assays to classify these cells as diseased or referenced based on an interest in a genetic cardiomyopathy. So to do that, we really thought about what defines cardiomyopathy at the cellular level. There are 2 major types that we have tried to model. The first is a concentric hypertrophy, which causes hypertrophic cardiomyopathy. In general, cardiomyocytes here are larger and thicker and have higher expression of this hypertrophy marker, NPPB or BNP. And then over here, we have a dilated cardiomyopathy, which is characterized by an eccentric hypertrophy, so larger, longer cells, and reports in whole hearts of higher NPPB expression. In real life, though, of course, those things are a little bit harder to replicate. So here is some data from Travis Hinson's group showing that at least for the gene TNNT2, if you have an HCM variant, you get higher NPPB expression. If you have a DCM variant, you get lower NPPB expression. So we really went back to the drawing board and said, okay, well, how can we think about modeling these 2 diseases? And we decided to split them. We decided to focus on cell size for hypertrophic cardiomyopathy using NPPB as a single readout. And then also I'll tell you about our dilated cardiomyopathy plans as well, which we are interested in particular in impaired differentiation or early developmental failure. So the phenotypes that have been explored in Ewan's lab successfully for hypertrophic cardiomyopathy have really been spearheaded by Chandru as well as by Hannah, who really led the mutagenesis for these experiments. And what this team showed is that you can actually use a microfluidic device, which basically takes cells in through an inlet, spins them around, and using centrifugal forces will actually move the larger cells to the outside of the channel, and then the larger cells to the inside of the channel, and then the smaller cells to the outside of the channel. And what ends up happening there is you can then split that channel so that you end up collecting all of the hypertrophy cells here. And this is just a picture showing you that MYH7 variant-carrying cells did end up more in the top outlet than the bottom outlet in this device. So Chenru has also used this to look at MYBPC3, which is another major cause of hypertrophic cardiomyopathy, and has shown a very similar pattern here as well. And at the same time, she has a fast speed camera which can take pictures and quantify the cell diameter, showing that for a couple of these phenotypes, we were able to replicate increased hypertrophy in that cell line. We've also used BNP expression or NPPB expression, as I mentioned previously, with FACS. And what you can see here is that for several pathogenic variants in MYBPC3, we do see a shift to the right toward expressing more BNP. And Yuta Yamamoto in Ewan Ashley's lab has really led this work as well in collaboration with Hannah. So what I'm going to talk to you about now, just to finish up, is our work on RBM20, which is actually a cause of dilated cardiomyopathy. So RBM20 was first discovered in several kindreds in 2009 and 2010 when it was reported. It's a very interesting gene in the cardiomyocyte because it actually is a controller of cardiomyocyte splicing, and it's fairly specific to cardiomyocytes. It's also slightly expressed in skeletal muscle, but otherwise, in general, is really targeted at the sarcomere of the cardiomyocyte. And the important part about that is that there are two major domains which are important for splicing. The first is the RS domain, which is important for nuclear localization and complexing with the rest of the spliceosome, and then the RNA recognition motif. Also, from prior work, we know that at least in the hearts of mice who have an RBM20 haploinsufficiency, we see an elevated BNP expression, which is critical for our single cell phenotypes that I just mentioned to you. And then we also see that the expression program of these cells actually looks a lot more like an immature cardiomyocyte, or when you differentiate them from iPSC stage into iPSC-derived cardiomyocyte stage, they actually look much less like an iPSC cardiomyocyte and a little bit more like an iPSC. And so we decided to capitalize on that. We decided to focus on NPPB expression as the readout for RBM20 cardiomyopathy, as well as impaired differentiation. So briefly, before I tell you a little bit more about our phenotyping, because it's going to involve some actual readouts, I want to go over how we're currently quantifying our variants that are associated with each phenotype. We do a low cycle PCR of the target region where we have landed our guide RNAs and created our edits. We then use a tagmentation-based library prep. We sequenced actually on an iSync, which has a relatively high error rate, as we'll talk about in just a moment. And then we have filtered on a variant frequency of 10 to the -4th, just based on the sort of shape of our data and fidelity with our controls. We then count those variants individually, and we have performed 4 to 5 biological replicates per region for the data that I'm about to show you. So how did we decide what areas of RBM20 to start with first? Well, we had done some work in humans where we took in vitae sequencing data in patients that had a diagnosis of cardiomyopathy and no other pathogenic variant, who had a variant in RBM20, and we plotted the density of those variants in RBM20 against what was seen in NOMAD. And what we found is that there were 2 regions that were highly enriched for variants associated with cardiomyopathy. Those included the RS domain, which we just discussed is important for nuclear localization of the splicing factor, but also this region in the glutamate-rich domain of RBM20, for which we don't actually know the function at this time. So coming back to our phenotype, the way that we've set up this experiment is that we perform our mutagenesis first by electroporating these iPSCs with the CRISPR-X and ABE8E plasmids along with the guide RNA library. We then FACS select them based on a fluorescence marker that we put in with the base editor. And then we culture the iPSCs that got the base editor. And then we use a basically Wnt inhibition and activation differentiation protocol all the way out to day 30. And then at day 30, we sequence. So the first thing that we do is we collect DNA just from the iPSC library so that we can know which variants were lost during differentiation. The second thing that we do is we sort the cells at the cardiomyocyte stage to find those cells that are not expressing alpha-actinin 2. Alpha-actinin 2 is an important marker of cardiomyocyte fate. So if you've gotten to the end of differentiation and you're not expressing this means you have not turned into a cardiomyocyte. And then we looked at the expression of BNP, which is our hypertrophy marker, as well, splitting this with the top 10% of BNP expressing cells by FACS as compared to the lower and normal BNP. So I'm going to show you some data now, and what you will notice about these distributions of synonymous and nonsense variants is that they are much more overlapping than I think anyone would like them to be in order to look at these phenotypes clearly. But I think that we have some ideas on how we can move forward with clearing them up, and we'll talk about that in a moment. So first, we looked at this iPSC to cardiomyocyte survival index. And what you can see here is that maybe we have a slight shift to the right on our nonsense variants in the RS domain, and we have definitely a shoulder here in the glutamate-rich domain, but not clear separation, as I had mentioned. We also looked at that differentiation index, which is the alpha-actinin expression, and we look a little bit better differentiated in the RS domain here, but at the lysine-rich domain, where, or rather the glutamate-rich domain, where we're having a little bit more trouble differentiating. And then lastly, our hypertrophy index of NPPB expression really didn't do a very good job of separating at all. The other thing to note here is that we have this long tail of synonymous variants, and we'll talk about why we think that is in just a moment. Also important to know about RBM20 is that its mechanism of action is thought to be dominant negative, where variants that are known to cause disease in RBM20 are located in the RS domain and actually cause the mislocalization of RBM20. What we know about that is that haploinsufficiency of the gene, although it's been shown in mice and rats to confer a very similar phenotype to the human missense variant phenotype, haploinsufficiency has not been proven to be a major disease mechanism in humans. So what we wanted to do here was jump the gun a little bit and look a little bit at the distribution of those PLP variants in the RS domain as well. What you can see is that maybe there's a little bit of a shift to the right in the iPSC survival phenotype, and then potentially also here in the iPSC differentiation phenotype. Not sure that we can say very much here about the hypertrophy phenotype in that case. So we have made maps, although we recognize that the signal to noise ratio here is fairly low. What we can say about these maps at this time is that we do recapitulate in the RS domain known pathogenic variants, as I just showed you, as well as others that we are currently chasing down with a low throughput method for contractility and calcium imaging. And then interestingly, actually, in the glutamate-rich domain, we see two regions that we're interested in. So the glutamate-rich domain itself is quite large, which is this red box here. But there was also that more narrow region that we had found in our clinical variant analysis that we were interested in. It turns out that these variants that are enriched here actually were seen in the clinical registry that I described to you, which is quite interesting and reassuring. And then additionally, we think that this region here may also end up being quite interesting once we've cleaned up a little bit of our error. Also, very interestingly, I think over half of the variants that were enriched by greater than 10-fold in the iPSC death group were actually missense variants at glutamate residues. And so there may be something important about glutamates in this region that we have so far been unable to understand because we don't have a crystal structure of RBM20. And then again, the second hotspot may be rather interesting. So lessons learned so far: multiple edits per cell make precise evaluation of missense variant function very difficult. And so what we want to do now is really work on how we can identify variants that were potentially dragged into the disease phenotype group, either because they co-occurred with a pathogenic variant or because they were actually part of a complex which was within the same codon. Additionally, we think that using narrow window BEs with highly lenient PAMs and more saturated guide RNAs may be another way to do this, because as we increase efficiency, we of course get more bystander edits. And then additionally, combining the populations that we have, so potentially combining the differentiation and cell death groups together in our analyses may give us more statistical power to better see these differences. We know now, I think, that BNP expression may not be a reliable method for variant effect mapping in DCM, or at least in RBM20 specifically, and that for HCM, iPSC-derived cardiomyocyte size and NPPB expression do seem to actually track with known pathogenic variants, and that we may be able to use survival and differentiation of iPSCs during the cardiomyocyte differentiation process as a sort of high-level phenotype, at least for beginning to map these genes at a lower resolution. And lastly, RBM20 variant causality is complex, with specific variants in the RS domain being dominant negative. Our ongoing directions are several. These are some amazing collaborators here: Lars Steinmetz from EMBL and Stanford, and his postdoc Kai Fenzel, who is visiting my lab this summer. Together, we're really working on the mechanisms of disease that may differ between those haploinsufficient variants that we talked about, as well as the dominant negative variants in the RS domain. And we're doing that in high throughput with single cell RNA sequencing. And then, of course, we're going to validate in low throughput. And I think that understanding what those highly enriched synonymous variants are will really help us to clean up our maps. So with that, I want to say thank you so much for your time, and I'm happy to take a question or two.
Questions in the room first. Is there anything online?
Nope.
I can't say anything. I'll just start off. I can't say anything. So obviously you did a lot of work to use the right cell type there. How do you balance the trade-offs of the challenges, I guess, of working with such a complex cell, but clearly the right one for the job?
It is such an important question, and I think it was really part of what we learned during this process. I think that now what we're thinking is, well, why don't we take ourselves back one step to a more functionally relevant assay that's bespoke or specific to the gene that we're interested in to start, and then we'll continue to work on this high-level phenotype now that we at least have it started. I think a lot of the work is going to be on the library preparation. Okay, thanks so much.
Thank you.